our second MASTS webinar, and I'm delighted to welcome Professor Alan Decho, who's visiting at the University of St Andrews as part of the MASTS Visiting Fellowship Scheme. Alan started out his academic career at the Connecticut State University, where he did a BA, then followed on his work in a number of laboratories, passing through Ohio University, then Woods Hole, and completing his PhD at Louisiana State University before moving for a year to CSIRO in Queensland, Australia, and then working for the Geological Survey for a period of years. And now he holds the Associate Chair and Director, is also for his sins Director of Graduate Studies at the Department of Environmental Health Science at the University of South Carolina. Alan and I go back a number of years due to our interest in biofilms and sediment dynamics. And Alan has studied the minutiae of the relationships between bacteria and is going to talk today on bacteria, chemical communications, and the implications and use of nanoparticles. So it's my great pleasure to welcome Alan Decho to this webinar on behalf of MASTS.
All right, thank you, David, and good morning, everyone. What I'd like to do today is give you an overview of the process of biofilm formation and hopefully relate it to a number of different topics that may be of interest to MASTS researchers. First of all, when we think of bacteria, we always think of a single cell or many cells floating around in a liquid medium, but really acting as individuals. What I hope to show you is that most bacteria do not do this. In fact, they tend to hang around in groups and, as we'll see, even act as individual groups. But they occur in the biofilm state when they attach to a surface as shown on the right, and this provides them a number of advantages and opportunities. It provides them protection. It allows them to acquire nutrients a little more efficiently and also localize different processes. So the many advantages to be gained are largely the result of what's called the EPS matrix, extracellular polymer secretions, that are produced by the bacteria themselves. So the biofilm consists of many cells in various shapes and sizes, surrounded by a self-secreted matrix of EPS. This allows them to exchange genes, localize their extracellular enzymes that they secrete so they're not lost to the surrounding water. But they also, over time, will bud off group parts of the biofilm to produce planktonic cells, which can then colonize other areas. But it provides, in general, a very stable three-dimensional environment as opposed to a one-dimensional environment. Now, I mentioned EPS is very important. What is it made of? It's made of a number of things: polysaccharides, proteins, DNA, lipids, but more importantly, it has a lot of different functional groups to it. This is a infrared spectra showing different functional groups that pop up on one type of EPS. And as you can see, it's loaded with things like carboxyls, amines, phosphates, so on and so forth. And this, in fact, is what gives EPS its many properties that are helpful to cells. Now, to show you what biofilms actually do, or what the microbes forming the biofilms do, here's an extreme example. These are called giant stromatolites on the right side, and there's a diver there just for scale. These can be 3 meters tall, but they are the production of bacteria, layer by layer, over perhaps thousands of years. Smaller versions are shown on the left side. But in addition to these giant dramatic macrostructures, we also see that in oceans they are involved in things like sediment cohesion, marine snow formation, the deep sea hydrothermal vents and the bacteria that live there, mineral precipitation, pathogens, and also things like animal symbionts, very important in those processes. In a more practical stance, we see water distribution systems, which are filled up with things like corroding iron and so forth. A lot of these processes are microbial mediated, at least in part. And we see the newer pipes made of other types of plastic-like materials. They become fouled as well, not to such a great extent. But of importance to MASTS and other oceanographic researchers is perhaps the biofouling process. It's a very dramatic cost, for example, to our U.S. Navy, somewhere around $200 million per year in added fuel costs just due to the biofouling of ship hulls. And the formation of biofilms is a first step in the colonization of other organisms that later are involved in biofouling, as we can see above: barnacles, protozoans, different types of algae, mussels, and so forth. The biofouling of sensors is also another process that's beginning to be looked at. We know now that the EPS matrix changes the optical properties of the surface that it's covering. It enhances forward scattering, and it forms what's called a biofilm gel effect. In other words, it has a different response to the sensor than would be if there wasn't a biofilm present at all. And we can see this in this diagram where the incident photons are coming in from the left side, just as an example. They're scattered in all directions, and a portion of these enter the instrument sensor. When we have a biofilm, there is enhanced forward scattering, so there are more photons going into the sensor under some circumstances and more photons being scattered under other circumstances. So the fouling just by a very small biofilm, tens of microns in thickness, could enhance or decrease the optical signatures that are being perceived by the instrument. And we call this the biofilm gel effect. In the study of marine mammals, we're now realizing that biofilms form on the surface of the skin of these mammals, dolphins, for example, lots of fish in addition. They're also very important in the gut bacteria of most animals. This is being studied to a wide extent in humans now because they're thought to be very important to maintaining our health, but also in perhaps many diseases as well. We always think of bacteria as just infections, but they're very important to our health and very likely to the health of marine mammals as well. So in short, biofilms are everywhere, and here we just see a simple salt marsh, but any surface that encounters water will likely have some type of bacteria attached to it, and hence a biofilm by extension. And so one important process we have been studying in a little bit more detail has been that of chemical communication among bacteria, a process called quorum sensing. This is where groups of cells release signals and are perceived by other cells, and it allows them to act as a coordinated group, and we'll see how this happens in a minute. It allows them to coordinate their gene expression, physiological activities, and also their behavior in many cases. It becomes a communication network, and it's a density-dependent process in many cases, not all though, and because the signals are released and they diffuse. So if the bacteria were floating around in the water, their signals would be released and simply float away. When they're in a biofilm, the signals can be more efficiently localized, so other bacteria perhaps can detect them and respond. So why target quorum sensing in our research? This is because it is likely a key step in biofilm formation and biofouling to a larger extent. We have begun work with Dave and Vladimir and others to try to examine how biofouling can affect the operation of different devices that are placed in the water. And so, in short, we want to know what is the role of quorum sensing, because this affects the biofilm and its ability to resist being removed and so forth. Quorum sensing is a multi-step process. First of all, the molecules are secreted by the organism, the bacteria. When they reach a threshold concentration, somewhere in the nanomolar range usually, there is a gene response, usually gene activation, but sometimes repression. This is followed by protein responses and hence physiological changes down the road. In short, as I mentioned earlier, it allows coordination of activities, and it allows the bacteria to now start acting as a coordinated group rather than a bunch of individuals out for themselves. There are many types of signals that are produced. We're focusing on what are called the AHLs. We'll see those in a moment. But there are many others, which I've simply listed a few of these groups of molecules here. We're using AHLs because they are very well characterized and we have good biological sensors. We understand their gene responses and so forth. So it provides a good platform for studying this process. The AHL is a pretty simple molecule. It's composed of a lactone and an acyl chain that varies in length from 4 to about 18 or 20 carbons. And the length and number of substitutions on this acyl chain will give it an identity. We call these a C6 or C8, depending on the number of carbons on this chain. But this also expresses the identity to the cell. Certain cells may recognize only C6 and C8 signals, or just C14s. Also, they may recognize several different types of signals, and they may regulate different processes in the cell. Responding to a C14 signal, just as an example, may trigger the production of certain toxins or the production of extracellular enzymes, while in the same organism, encountering a C6 signal may cause the bacteria to up and leave the biofilm. There are many roles that these signals have to the cells, and it depends on the species and their genetic makeup and how they respond. So I guess what I'm trying to say in a roundabout way is that it's a very complicated process. You could have different combinations of signals, in effect acting like different letters in words. Different combinations will change the meaning of many of the signals to the cell. So a lot of the military are studying this bacterial type of signal because it's a very simple but efficient way of information processing. And so they're very interested in that. Now, how does it happen? We have the signals up top, the AHL signals out in the environment, diffusing around. They then cross the cell membrane, diffuse into the cell. Sometimes they're actively transported in, where they encounter a receptor protein, shown in green. And once the receptor proteins and AHL contact each other, they both change shape. And at this point, the protein then begins moving towards the DNA operon, where it attaches and begins activating the gene. And so this is a very straightforward example of how this works. Sometimes the proteins and signals have to form dimers, and even trimers have been detected. So it certainly has a lot of more complicated pathways as well. Now, bacteria in nature, when we look at them, we tend to see that they form groups, which would suggest they might be quorum sensing. On the left is a cross-section taken with a confocal microscope of a microbial mat from a stromatolite. Dave Patterson and myself have worked on these systems, and shown in, it's very colorful just because of the fluorescence of the confocal, but the purple are sediment grains, they're very round, but the things of interest are the gold masses up top, which are sulfate-reducing bacteria stained with a fish probe. The red stringy things are cyanobacteria. I think what you can see is that it's packed with bacteria. We'll see there are over 30,000 different types of bacteria here. But when we look up at these close up with the confocal, we start to see the individual cells. And this, in fact, is what we see are clusters of cells very close to each other, probably signaling each other and communicating very rapidly. And just a blow up at the bottom of one of these clusters. When we look at any type of surface, we will see the same clustering of bacteria. And what becomes important in the chemical signaling process is who is communicating. Are they simply communicating within a single cluster, or are clusters communicating with other clusters? And if so, what is the calling distance that signaling can occur? How far can this occur? Remember, each of these bacteria shown here are approximately a micron in size. So these are relatively small distances, but when you put in the EPS matrix that we cannot see here, it could slow diffusion significantly. So cell clusters appear to be ideal places for chemical communications. Can we actually find signals there? And to do that, we extract the sediments in this case, and using a range of organic extractions, we pull these out, purify them, and then run mass spectra on these. And we can see a natural mat has many different peaks on the bottom chromatogram. At the top are standards of different sized AHLs that I mentioned earlier, a C6, a C8, a C10. And when we get the same retention peak, we could then isolate that peak with the mass spec and rerun it under a higher energy and break that molecule. If it breaks into two pieces, the acyl chain and the lactone at 102, and the acyl chain of varying length, then we have pretty good confidence that in fact this natural molecule is a signal. It's behaving exactly the way a standard signal would under those conditions. We never know for sure, of course. And on the right is a bioassay showing that the signal activates a certain type of bacteria that respond very well to AHL signals. The dark blotches are the bacteria reducing a colorful compound. When we extract the signals over a day versus night, as shown in this graph, the nighttime in black, the black bar, the daytime, the blue bar, we see significant differences in their concentrations. And this is likely due, or in part due, to photosynthesis. The pH of these mats is very high during the day due to net photosynthesis, and this produces an alkaline pH, of course, and that is destructive to the AHLs. So part of these AHLs, C10 and C8 in this case, may be at reduced concentrations during the day simply because they're being hydrolyzed by the alkaline pH. At night we see higher concentrations. Another possibility, of course, is that they're secreting more at night than in the daytime, but laboratory evidence thus far at least does not indicate that. Another challenge that signaling has in many places that bacteria live in biofilms is desiccation. Remember, bacteria are exposed to many environmental challenges such as salinity changes, pH changes, UV temperature, and we find that in these microbial mats, when they are hydrated, as shown in cross-section on the left, we can see different functional groups of bacteria, the diatoms on the surface, cyanobacteria underneath, sulfate reducers, and the other groups. When these same mats become very dry, as shown on the right, they have a lot of salt, which forms concretions on the top. The bacteria are completely dried up and remain so for months at a time. Yet, when water is added, within an hour or two, we can detect quorum sensing. This seems to be too rapid for cell production of signals to reach those concentrations from what we know in the lab. So we have to suspect that the signals themselves are surviving in the dry mat. These are pretty delicate molecules, but yet they're surviving somehow. And so we tried to set out to see how this might occur. And we are coming to the conclusion, based on data, is that there's a formation of a glass, what's called a glass within the EPS. This is where you get an amorphous structuring of molecules that allows a protein, for example, to be protected from denaturation. Kind of similar to the way a chaperone works within a cell. And it's realized now that glasses are used in the food industry to preserve proteins that are in food, which provide flavor or texture to the food. So anything in a box that you buy will likely have a glass that has proteins that are important to the food embedded within it. We're finding trehalose in very abundant amounts within the EPS, and this is likely one of the things, at least, that may be forming the glass and hopefully preserving the AHLs. We have done NMR spectra on these, where we're looking at the trehalose by itself, the AHLs by itself, and the two together. And they're starting to suggest that, yes, several trehaloses bind to a single AHL. They kind of swing around it like horseshoes and perhaps protect its shape. We're trying to have the chemists model this, but apparently it's very complicated. And so when we create EPS glasses in the laboratory, we can expose the signals to high temperatures, to very alkaline pHs even, and they're able to survive and retain their biological activity. So that's very interesting to us because in nature, bacteria are exposed to this desiccation process in many, many places, and it allows them to kind of just quickly begin metabolizing once they're rehydrated again. And this is just another view where we are continuing our studies of microbial mats. I mentioned stromatolites. We're also working on hypersaline mats, as shown here. They're very orange in color due to the prevalence of archaea, but there are over 30,000 different types of bacteria and archaea in these mats. Very, very abundant, and the question immediately arises, how do they know who they're talking to when they're sending out signals? 30,000 different types of bacteria all sending out signals. How do you control the signaling and not have too much interference and static? There are also novel antibiotics and anti-cancer drugs that can be found in such mats, and they're used as a model for understanding early life or extraterrestrial life. Now, this brings us to another research question that is related, and that is, how do we control or manipulate biofilms and hence the biofouling process? One possibility is to use nanotechnology. We'd like to see if we could manipulate surfaces in a way to either reduce the formation of biofilms or to get rid of biofilms once they're there. And in targeting the chemical communication process, this provides perhaps a very good tool. Very briefly, nanoparticles are particles that are on the scale of about 1 to 100 nanometers. Remember, a bacteria is about a micron or 1,000 nanometers. So particles at this size seem to offer unique physical-chemical properties, and they tend to act different than larger particles, even of the same composition. And so what we're trying to examine first is how will the EPS matrix inhibit or enhance the movement of nanoparticles through this matrix. Remember, the EPS, as shown in this atomic force micrograph, is a series of molecules intertwined in different ways. And the dark spaces in between the lighter molecules are water spaces. And this is where diffusion occurs through the EPS. So you can see how it could be slowed in any type of molecule, small or large, trying to move through this EPS matrix. And EPS varies in how dense it is or how loose it is. It could range from totally dissolved molecules floating in the water. A lot of the DOC in oceans is EPS, to very tight gels that can't be penetrated by even antibiotics. And so the bacteria are manipulating this matrix to some extent, at least, to protect themselves and afford themselves adaptations. And so we want to understand how nanoparticles, especially with surface charge groups, could interact with EPS. What about the nanoparticles can affect their binding to and penetration of the EPS matrix? And maybe so, how could we manipulate the coding on nanoparticles to enhance this or increase this specificity? That's what the protein corona refers to. This is an area being studied a lot in medicine at present. But what we are doing with chemists at this point is trying to create nanoparticles that can disrupt the quantum sensing, and hence we call them quantum sensing disruptors. And what is shown in this schematic is a nanoparticle with raft-like molecules extending from the nanoparticle. And these can be synthesized in different sizes. And then we attach what are called cyclodextrins, shown in the bottom center, which are round, kind of donut-shaped molecules with a hydrophobic core. And these bind AHL molecules, shown on the right, which are signals, very well. So we attach the cyclodextrins to these long raft molecules, and we could attach up to 30,000 at present onto a single nanoparticle. If the raft molecules coming off there become too dense, it's kind of like a hairbrush, it's impossible for the AHLs to reach the cyclodextrins. So we have to manipulate these to only a certain density and not too dense. And when I say we, I mean my chemistry colleagues. It's a very involved process. But what we hope to do is create nanoparticles loaded with cyclodextrins, which could bind the signal molecules. And so if they could be implanted or migrate into a biofilm, they could begin binding the signals. And we're looking at the binding strength of the AHLs on these cyclodextrins, which are on the nanoparticles using NMR at present. And we're starting to get some very strong binding with modifications of the inner core of the cyclodextrin. And so we're hoping to get more specificity in the binding of cyclodextrins, modified cyclodextrins with AHLs. And then what we hope to do is to attach other types of raft molecules, perhaps longer than the cyclodextrin-containing ones, that will help facilitate the migration into the biofilm. They'll help the nanoparticle slither by the EPS molecules and not get stuck. And that is a big problem. But if all works well, we use a luminescent bacteria which produces light when it's quorum sensing. Vibrio fischeri is the name. And you can see on these otter plates, especially on the right, those are bacterial colonies which are producing AHLs, and they're quorum sensing, and hence they're producing light. When you put them in a culture flask and they're dense enough, they produce lots and lots of light, as shown here. Someone's holding this up, and it produces a lot of light. You could read a newspaper with it. And so what we hope to do is, when we get our particles working correctly to disrupt quorum sensing, we'll have all these lights being produced by the bacteria. We'll add the nanoparticles, and if all works, the light should go out. And then we'll want to verify this enzymatically, or actually at a genetic level using PCR. And so one of our hypotheses that we're trying to explore further is how the density of the EPS matrix and the pore space size may affect the ability of nanoparticles to enter and penetrate the EPS and biofilm. And using very simple models in the laboratory at this point, we're using different size nanoparticles with different surface functional groups, and we're manipulating the density of the EPS gels, and then trying to actually measure the sizes of the water spaces in between the molecules using atomic force microscopy. And if we could create the gels very consistently, at present we could get different size pore spaces on average, and using small enough nanoparticles, we would want to see if they could penetrate into the gel more efficiently than if the pore spaces are too small. It seems reasonable right now, common sense, but nature works funny, so we're just trying to take it one simple step at a time. And so really the challenge of the biofilm is the chemical communication. Certainly, can we get to it, perhaps shut it down, maybe even specifically just shut down certain genes in a bacteria? This could affect biofouling. But before we do that, we have to understand much more about the EPS matrix. It's been called the black box of biofilms because, or the dark matter of biofilms, because it's not very well understood relative to cells. And so we're trying to see if we could manipulate nanoparticles to penetrate the biofilm, as shown on the top left, versus getting stuck at the top. Biofilms, remember, in nature are very heterogeneous, I think is a simple way to put it. They differ in their density, their composition, their even movement. They're very complex adaptive structures. And so when we start examining them in nature, there will likely be a lot of variability to deal with. And so for this reason, we're looking in the laboratory at very simple biofilm systems, but we're concurrently trying to work in the field examining the same processes in natural biofilms. And with that, we hope to continue our study, and at this point, I would like to open it up for any questions that some of the listeners might have. Thank you.
Thank you, Alan. While John is working with the technology there, I'll start off. When you have a drying biofilm and you create the glass-like state, that suggests that the signal that's being preserved is the signal that was there at the time of the drying. Why would that be the appropriate signal to be there when the hydration starts again? I'm just curious about whether a different signal would be necessary, or you think it's just what's it doing, that signal? Why is it an advantage?
Well, we suspect that the bacteria are under stress. They're getting ready to dry up, basically. And so they're probably signaling each other, having to do with the desiccation that's occurring or the very high salinity that's occurring. And so it may be to activate genes to produce more internal chaperones or produce more trehalose. The genes for trehalose there are many different forms of them that we're finding in the natural MASTS. So
I think that's partly my point. If trehalose is a protective mechanism, so I can see that you create the trehalose as the system is drying, but then as it rehydrates, you've still got that signal there, which would seem to me to create more of the thing you don't need anymore because it's rehydrating.
Yeah, the only way we could reason what they might be doing, we have to think like a bacteria, I guess, is that as they're rehydrating, they're going to be exposed to very high salinity conditions. And sometimes this rehydration involves a brief rain, which gets them wet, but then it dries within a couple hours again. Mm-hmm. So I think they're getting ready for more stress when they wake up. I don't know. I guess it's the short answer. Yes.
If you succeed in getting nanoparticles to inhibit biofilms, are you not worried that you're just creating a major new class of pollutant? Because if it gets into the water, it's going to affect the gut health of everything that lives in it, including ultimately ourselves.
Certainly. That's a big area of concern. I think we're already there in that nanoparticles are used for so many different things in industry. I think it's predicted to be a $15 trillion business in about 3 or 4 years because they're added to everything from our socks to orange juice, all sorts of things, suntan lotion. So we're already putting a lot of nanoparticles in the environment, and there are lots of natural nanoparticles produced. But yes, that's a major concern.
A formulation that specifically affected biofilms you think would be particularly problematic?
We don't know if it would be any more problematic than, say, a silver nanoparticle, which is toxic to everything. But that's something that will have to be tested very stepwise. One of the things we'd like to do is carry antibiotics into infections in the human body, and it's at least 10 years if everything goes according to plan, which it never does. You have to make sure they're not toxic to the cells, then to rats, then to humans. It's a long, long process.
Can we invite any questions from the audience at large? Are there any questions from the hyperspace? Okay, in that case I'll continue at the moment, which is just to follow up on Ian's point. If the nanoparticle adding simply disrupts a quorum sensing molecule, is that process reversible?
Yes, the bacteria should be. It's simply acting like a sponge. So as soon as it gets saturated, quorum sensing will begin again.
Yeah. So that actually is something disrupting an environmental process may not be as nasty as, say, a silver particle, because once that nanoparticle is saturated with the signal, then that's it out of action. Is that right?
Yes. In terms of infections, though, it's advantageous to inhibit quorum sensing because it doesn't kill the bacteria. It simply shuts down some of their processes. And if we could shut down what the toxins it produces that makes us sick, the virulence factors, then the bacteria that are nasty to us could suddenly just become okay sitting there. They're not doing anything bad to us anymore. And we have a lot of examples of that with E. coli. If it's in the wrong part of the body, it'll kill you. If it's in the right part, it produces essential vitamins. So it's
So John, I'm not quite sure of the technology. Very early webinars. Oh, sorry, we have a question, I think. Ah, right, yeah, okay. There's a question come in from Haideng, who wonders what happens when the biosynthetic pathways of HLS, presumably production, is knocked out.
well, they have mutants that we use in the lab where they don't produce AHLs. And so they've already done that. In nature, I would imagine this would be a problem for the bacteria, or perhaps it would not, because what they would become is what's called a cheater. They're able to perceive the signal, but they're not investing the energy to produce it. And people have done numerical studies where as long as the cheaters remained low abundance, they could survive in the population quite well. They're kind of in the background, picking up the scraps, using what the other bacteria are producing, signals in this case, or reaping the benefits from it. And when they get too abundant, then the whole system crashes. So I'm not sure if that answered your question.
So if you have these populations that can't produce AHLs, what's the difference, in terms of their growth and functionality? How can you keep them alive or not?
They should be able to. They're able to grow. The AHLs in most cases simply allow them to coordinate some larger activity that's better done as a group. Mm-hmm. So they should have all of the functions they need to live just in a culture flask.
But then if you get a mixed assemblage where you have too many, as you call, cheaters, then something falls apart.
Yeah. That it would seem like the biofilm itself would be weakened, and that would be selected against.
So it doesn't function competitively as a biofilm anymore.
Right.
So if you could do that in terms of having some kind of way of delivery of a mechanism for turning off HL synthesis, then you would have a potential control of biofouling.
Yes, and in fact that's being, we're starting to get interested in this in other groups as well, in attaching microRNAs to nanoparticles, and if you could get them inside the cell, you could actually turn off specific genes or specific proteins, and that could be one of them. Okay. It would be a very specific way of controlling one process at a time.
Okay. So becoming quite sophisticated and targeted.
Yeah. At least on paper.
Okay, thank you. Any further questions? We're at the end of our time allocation. So in that case, I will round it up and say thank you very much, Alan, for taking part in this.
Thank you.